thanks for inviting me. So I'm here to talk about the biotech devices that I've been able to work on, fortunately, over the course of my career. I'm hoping I can get the slides up because I just changed it. There we go. Okay, but first I'd like to tell you how I got into the biotech field. So I was raised in a poor family with 6 brothers and sisters in Cincinnati, Ohio. And when I was 6 years old, our entire family was riveted in front of the black and white cathode ray television set, watching the first manned space mission going to the moon. And when Neil Armstrong first stepped on the moon, I was amazed. And I thought, okay, I want to do something great like him someday. So I'm going to be an astronaut. And it ended up, over the course of the next 10 years, I worked extremely hard in school, both in academics and athletics. And I was able to get to an excellent high school, which was actually very similar to Gunn High School. And then I finally was able to work my way to going to the top university in the United States for aeronautics and aerospace. And that ended up being Stanford University, where I could actually try to be an astronaut. The nice thing about Stanford at that time is 1 out of every 60 graduating students did get up into space. But then tragedy struck. In the the end of my master's program, the space shuttle blew up, the Challenger. And I was devastated, and pretty much all of my colleagues were as well. And I had to decide at that point, now that manned systems were going to be on the decline, what was I going to do with my career? Because I had focused so much energy and desire into going in that direction. Luckily, having a mechanical engineering and aeronautics degree, it gave me the skill set that I would need to change my career and find something else that I would be passionate about. And within the next few years, I worked my way into software development and creating devices for various instruments, and then luckily worked my way towards the biotech field. And I'm going to talk about the 2 revolutionary devices that I was able to work on. The first one, okay, I'm going to skip that. There we go. So the first device was the world's first high-throughput DNA sequencer. So I think everybody's heard about the Human Genome Project, which was started in the late 1980s. And the goal was by 2005 to have it completed. This system was developed in the mid-1990s. I was a software manager. And during that time, sequencing DNA was a very tedious process. So by automating the system, we were able to improve things tremendously. And I'll go into some details. So the hardware of the system is extremely cool. So in here, these are capillaries. So imagine taking a huge tube of glass and extruding it so thin that it's now thinner than your human hair. And then putting a polymer coating, a rubber coating outside of that glass, so that now you can almost twist it around into a knot and the glass won't break. So by using that technology and creating 96 of these capillaries, you were able to load the DNA sequences that you wanted to run through the capillaries. And a gelatin material was then moved through these capillaries past a laser detector that would scan back and forth about 3 times a second, collecting all the data for the ACGT base pairs that the genome sequences needed to get. And pretty much this device automated an extremely tedious process. And let's go into it. So the software that I developed while you were running the instrument looked like this. So you had all 96 capillaries. So I'm trying to give sort of a global picture of what was going on the instrument. And as time went on, each of these rows would slowly work its way down, and you'd see different barcodes of the various ACGT base pairs being displayed. And then if you clicked on any one, all of a sudden you could see a chart of those specific sequences as they were going by from the beginning to the end of the run. Now, why did we come up with this design? Because the capillary system was this unique new system that nobody had seen before, but we had to relate it to something that the industry was already comfortable with. And that was 20 years earlier than when they first started doing DNA sequencing. They used to radio label the base pairs that they wanted to detect. And what would happen is you'd put some material in the top, run this through an electric field, and these would slowly move down through this slab gel. So imagine 2 pieces of glass that were approximately a foot by a foot. You had to tape the outsides. You had to put gelatin material in there with spacers. Then you had a comb at the top where you would inject this DNA material. Extremely labor intensive. That's the way the field was before our system came along. Then, with some technology improvements, and instead of doing radio labeling, they were able to start using fluorescent dyes, which were a lot safer than having to deal with radioactive equipment. And they were able to work their way up to 36 wells. But again, this was still a big glass area with gelatin material. You would go and inject the DNA that you would want to run. It would run for a certain period of time, and the fragments, smaller fragments, would move faster than the bigger fragments that were labeled, and you could detect each of the ACGT base pairs as it went by. But there were a lot of flaws with this system. But what we realized is that's what our customers were used to. So we had to go and simulate something that they were familiar with so that they could more quickly adopt this new technology and get to the information that they needed. What I found absolutely fascinating about this project is once we deployed all these systems out into the field and everybody understood it, the entire decade before, the amount of DNA in the human genome we could sequence, we were able to do in that first month of getting this system out. So it really advanced the field, and as you can tell now, genetics is really at the heart of the new biotech revolution. Once that system started being deployed, I'm sorry why this is so fuzzy to look at, but I worked for the company at the time that was the best sequencing center in the world, and it ended up being Insight Genomics. And this was right before Celera started coming up on the private sector for genome sequencing. And what they wanted me to do was put 48 of the systems that I was a software manager together into one collective system so that as efficiently as possible, they could go and collect all of this information to then put it together for the Genome Project. So imagine a production floor. So the way I look at any software problem is I try to zoom out to 10,000 feet, tell a story, give the big picture, and then describe to the customer in ways that they understand what they're trying to accomplish. I try to give them a user interface for that. So this is a production floor there were a bunch of aisles where the technicians could go in, and then there was a big lab bench which had 8 instruments, each of these being the serial number for the instrument. And what would happen is I made this an HTML page in the late 1990s when HTML was relatively new. And we projected this on a screen almost like you would see now in our production facility. And every minute it would update. And it would go and it would pretty much show how long the last, what was the last run, who ran it, what's the ID number for the plate that you're actually sequencing, what was the previous run, what were the read lengths for that instrument for the runs, how many fails of the capillaries, because that was a big issue we had to detect. And whenever anything went wrong, the system would automatically notify the service people or myself what was wrong, when it was wrong, so as quickly as possible we could keep this system running. So it was just a marvelous project to work on, and it really led the way to where the next-gen sequencers are now. The second project I wanted to talk about is the world's first droplet digital PCR system. So I worked at a startup company called QuantaLife, and joined the company 3 years ago. And when I was first interviewing with them, the co-founder showed me this. And I was absolutely amazed, because what happens is, imagine a teardrop, and breaking that teardrop into 2 million unique droplets, each of them of the same volume, where you could put in 1 cell, or you could put in 1 genome's worth of DNA. So now you could do an individual experiment on every single cell. And when I saw that, and I could see how consistently they could do it, how cheap it could be done, I said, please, let me sign up, let me do this. So my job here was to be the software developer for the instrument control and the analysis of the software. And the inside of the instrument was kind of fascinating. This is for reading the droplets. So again, you'd have a 96-well plate down here, where in each of the wells you'd have 20,000 of these droplets. And then this needle would go down into there, suck up the 20,000 droplets, move it fluidically through this system. It would go past the detector, and the detector would go and read those droplets as they were uniformly flying by at extremely high speeds through this system. Come on. The software for controlling the instrument, I tried to make it as intuitive as possible. So this was actually, and I don't know how well you can see it, but this was a simulation of a 96-well plate, kind of like an Excel spreadsheet, where you had the rows and columns of your plate, and you could go and click or double-click on each one of these cells and input very efficiently the 9 or 10 pieces of information you wanted to track. And for this specific system, you were able to do 2 experiments in each droplet at the same time. So you had 2 different fluorescent dyes that could be tagged to help you determine whether or not that DNA contained a specific sequence that you were interested in. The analysis side of the software, especially when you have a brand new device, it's absolutely critical to have a manual system where the user can interact as much as possible to get the exact results that they want to see. So in our case, you could select, so again here, 96-well plate simulation so that the customer is familiar with it. This was an Excel spreadsheet fully embedded into the program, and here was a huge charting system of all of the individual droplets intensity readings. And you had various tools where you could actually go and circle a certain section, say, well, these are all my positives, these are my negatives, or you could just draw a cursor band to actually separate out the sections to make it clear that these are all my droplets where the DNA occurred, and these were all the conditions where it didn't occur. And ultimately, the end game was, what is the concentration of droplets that contain the DNA sequence I'm looking at, and how out of the total number of droplets. And when you have that on the individual genome level, it's extremely powerful, because now you can detect that very first cell or that first condition for HIV in your body or in your bloodstream. So one of the collaborators for this project was Sangamo Biosciences, and I'm going to show you a short video about what they do and how they've used our system.
So the primary objective at Sangamo right now is to apply the zinc finger protein technology. I think this is the only technology that allows you to work at the DNA level to knock out the major co-receptor for HIV virus, CCR5. And then we'll subsequently infuse these protected cells into the patients, which hopefully will provide them sufficient immunological benefit that will provide a functional cure in these subjects. The QX100 technology is really a very useful tool for us in that we can use it to analyze the changes or fluctuations in the patient's HIV reservoir, allowing us to monitor what's going on once we have reinfused these cells into the patients. Or once we acquire the QX100, where we can subsequently repeat our measurements and see the same changes over time, we're a lot more confident in what the assay is telling us and giving us something that's biologically real and relevant. Because if we're using a technology that generates data that we can really trust. The way the droplet help is that when you break up a sample into 10 to 20,000 droplets, essentially within each droplet you're doing a single PCR. So instead of doing one PCR, you're essentially doing 10 to 20,000 PCR, where only a fraction of all that 10 to 20,000 drop will contain your positive copy. So essentially what you did is you dilute out the background level of DNA, increasing your positive DNA to background DNA level to a point where you can more accurately determine the positive template that you're trying to amplify. Fairly straightforward. It's not a very complicated procedure. There's a simple protocol that you follow. I think the first experiment we worked with our collaborator on this technology, we saw the result and we were amazed at how accurately and reproducibly they can measure low copy number events in a sample of genomic DNA. So the QX100 really is a godsend for us in a way that we can now more accurately measure the HIV DNA level?
So what I've learned through this entire process in the biotech field, especially for the seed companies, is you have to follow your passion. So originally I thought, okay, I want to be an astronaut. Well, life doesn't always work out the way you want it to. But having that passion and driving it into another field where you can help mankind in some way was very rewarding for me. Working hard, it's so much easier to work hard on something you believe in. So try to find something that you believe in, that you're willing to put in those hard nights and the all-nighters. You need to be proactive, because what I found too is if you don't direct your course, what ends up happening, even if you're a very smart person, is somebody else will try to guide you into what they need done or what problems that they want you to solve. And I think if you're proactive in your career, you're going to be able to push the limit a lot more and make a bigger impact. The teams that you find and make for the projects, it's critical that everybody believes in the cause. So in my case, both of these were relatively easy to be passionate about. One was the Human Genome Project, speeding it up drastically. The other one is starting a whole new industry where all of a sudden now you can measure each individual genome and do reactions on that. The other thing that I think is very important specifically to the software world is software has to tell a story. And you may think you know the answer before you get started, and in a new device and a new technology, you don't. So you have to rapidly iterate and get that user interface and get that technology out into the customer's hands. And as soon as they see something wrong, something doesn't work, you now understand better how the customer thinks, then you have to go back and quickly put something else out there. And in the software world in general, I would say that that's the direction that the technology is going. So that's it. Thank you for letting me share with what I've done.